last year, George and I gave a talk at one of these Odyssey community calls where we tried to highlight strategic opportunities that we thought we wanted to showcase with the community as an opportunity for how we could all come together and collaborate. And we listed like 4 big ideas that we wanted to throw out there as things that we'd like to go after as a group. And actually, idea number 1 was how often. And the basic premise of this was that we think that there's an opportunity to conduct large-scale characterization of the incidence of outcomes within specific populations. And we posed basically what it would take to do this and what are some of the open scientific questions to be able to handle this correctly. Now, a lot of this is motivated by the fact that as a community, we've already made really good progress on learning about the scientific integrity of producing incidence rates and how we can do this across our network. We've published papers that provided incidence rates, specifically providing information to the EMA and the FDA as it related to the COVID pandemic and adverse events of special interest for vaccines. We've published papers that looked at the methodologic issues about how to compute incidence rates and how they are sensitive to various characteristics that can go into the analysis. And Erica led the largest Odyssey network study that contextualized adverse events within multiple populations. And so we've demonstrated this could be done at some level of scale. And what George had proposed last year, and we're going to go through today, is how we can actually take this idea even further to produce a substantial resource that the entire community could benefit from. So what we're going to do today is George is going to kind of tee up and motivate this problem. What I'd like for all of you to be thinking about is thinking about input that you could provide George and use the chat to post your questions, your comments, and your concerns. And then at the end, we're going to specifically be opening up a poll everywhere to be soliciting specific input from you about how, where you think we should be focused as a community and what contributions that you all can make. So without further ado, George, I'll hand it over to you, and I'll be happy to drive the slides for you. Oh, I should also mention, also in our community, we've developed multiple package standardized analysis packages, including the cohort incidence package, as well as the incidence prevalence package. So we've demonstrated the technical capability of what George is going to talk about, and now it's the opportunity to put this full throttle to take advantage of a real opportunity to generate reliable evidence that can be meaningful for the broader community. All right, George, all yours.
Great, thank you so much, Patrick. Hi, everyone. And Patrick, if the construction out my window becomes objectionable, let me know, and I will be running to your office across the way. So let's see how that goes. So we're talking about starting off with the incidence of all effects of all drugs. If you ask your doctor what are the important side effects of the drugs that they use commonly, they'll know the ones that were most suspected in the first place are the ones that are most common and they show up. They probably don't know all of them, but they know many of them. But if you then, even for the ones that they know about, if you ask them, well, how often does that happen? What's the incidence? They usually have no idea what the actual incidence is. They have a vague idea, is this something that's worrisome or not? And if they go to look it up, the evidence for that is pretty sparse. To calculate incidence rates is a lot easier than doing causal inference. So we can find out the incidence of things on large sets of hypotheses. For example, a very large set would be every drug on the market in the world. And for every outcome, every possible effect in SNOMED, that would be a large set, that cross product. Because incidence is pretty easy to calculate, we can actually do that. And we can tally it for every pair. So now, how can we use these rates? Well, if the incidence of a side effect given a drug is low, then I know I'm set. The drug might cause the side effect, it may not, but if the incidence is low and the side effect is not too serious, it's not death, then I'm probably pretty safe in giving that drug in this context. If the incidence is high, then I don't really know what to do. I have to look out for it. So I don't know if the drug is causing it just because the incidence is high. It could be something else. However, it's still useful information. If given that I've gotten this drug, I have a high incidence of something going on, say renal failure, it's something I need to have on my mind, even if it's not caused by the drug. As I said, it's feasible, fewer assumptions than a causal inference. But even with all this simplicity, it's still pretty hard. So what is incidence? Well, there's some entry into the cohort shown on the top blue arrow. There's some baseline observation to the left, the observation start period, where we may have some qualifying criteria. Then after the cohort entry, there's some time at risk, where we're seeing if the outcome occurs. You exit the cohort, and then there's the end of your observation period. In other words, the end of your record. And when we study incidence, we need to make some decisions based on these factors. Next slide. The incidence, now there are two different things we can measure. The incidence proportion, which is kind of the proportion of people who have an outcome, say a side effect, within some given time window. And there's the incidence rate, that is per unit time, what is the rate at which I'll see that effect. There's an important difference is, and if you look in the literature, they're reported both ways. The incidence proportion, I need to decide ahead of time what time period I'm interested in. For the incidence rate, because it's a rate, I can adjust the time period for different people, as we'll be talking about that in a future slide, and still have a consistent answer. Next slide. As I mentioned, incidence rates are not causal. So say in a drug like warfarin, which is an anti-clotting drug, there's a baseline risk of stroke for a person off warfarin, and that's the dark blue. If you take warfarin, it might induce a hemorrhage in your brain and cause a stroke. Of course, the reason you're taking the warfarin is because it might avert a stroke because it stops emboli from going into your brain. And the hope is that the green is bigger than the red and warfarin is beneficial. The net incidence is going to be the light blue or teal on the right, which is the observed incidence. So notice what we really want to know is the causal effect of warfarin, which is the attributable effect, which is that bracket on the right. What we can see, though, is the light blue on the right. But that's still an interesting rate, even if it's not causal. Next slide. So let's go through some of the things that we have to figure out. Next slide. So how should we define the target cohort? That's a new user of a drug. So generally what we want is the date of first exposure to the drug. Then that raises questions like, well, what's your look back period to decide this is a new user? If we look back 10 years, and most people don't have 10 years of records, we'll have a very small sample size. If we look back just one day, then we're not going to get anyone who had previous prescription for the drug, since the period is 30 days. So we need something in the middle, maybe 1 year look back to see if someone was on the drug before, if we want a new user of the drug. Having a new user of the drug tells us the incidence of these things, and gives us a rate that's a little bit closer to what we're interested in. It's not causal, of course, because it's an incidence rate, but it's not so complicated by prevalent use. Next slide. How should the outcome be defined? That is the side effect. One way to do it is to look for a first time ever in your history that you had this diagnosis, because we're not really that interested. So if someone starts a drug and they have a long history of heart failure, are we really interested in their incidence rate of heart failure after they start the drug? We may be, but if we're kind of interested in what happens in the context of the drug, recognizing once again it's not fully causal, we may want the first one. Or we may just want to have the first incidence of that side effect since you had the drug, but it's okay if you had it before. A clear example of that is some kind of infection, something that you expect to occur many times in a person's life. You wouldn't want to rule out someone who had a URI in the past, from having another one in the future. So that is something that you would not want to have the first ever incidence, perhaps. Next slide. And then the time at risk, are we talking at 30 days for short-term things? Anaphylaxis is going to happen in a day or two. Or do we want like a year, which is our normal? Or while they're on treatment for the drug? Or do we want it forever until we finish their observation period? There are arguments for each one. But I won't go through in detail those arguments right now. Next slide. Now, if you have incomplete time at risk, what do we do with that person? So in other words, let's say you say I'm going to look at 1 year, and I have a person in my database where their time ends at 6 months. Should I include that person in the tally, or should I exclude them because they didn't make the full time at risk? Well, it turns out if I include them, then I'm gonna have an incomplete follow-up time, and I'm gonna have a lower bound of the absolute incidence. If I include only persons who had the full time at risk, then as long as the rate is roughly uniform, I'm going to have an upper bound on the true incidence rates. So what do we do in OHDSI when this happens? Usually we do it both ways and see it as bounding, as more or less bounding criteria, although strictly speaking, it's not necessarily an upper bound depending on the change in the rate. And then what metrics to report? I already talked about proportion versus rate. One thing we like to do is compare what's in the literature, and there's a mixture of things in the literature. Do we report 95% confidence intervals for our calculations? Well, we have long experience that if you just take your sample size and calculate 95% confidence intervals, they're way tinier, way more precise than the actual variance we see due to systematic error. One approach we've taken is that we look across the databases in our network and see the differences among the databases, which capture some of that systematic error, perhaps not all of it, and is a better measure of the overall variability than simply calculating the confidence interval based on sample size. And then what data should we use? And we always try to get a broad representation across our network as possible. Next slide. Just to point out that uncertainty on these rates come from a variety of things: genetics, environment, bias, problems in the ETL, sampling error, and confounding, and so forth. So we really want to capture this variability. Next slide. So how are we doing today in the literature with estimating incidence rates? Well, for the majority of incidence questions, there's no readily accessible evidence available. What happens? We have, for new drugs, we have randomized trials. They do a subset of side effects they think will be important, and they have a small sample size, so they can't capture the rare ones. And then we do observational studies after the fact, although they're decided by the researcher and the editor whether they publish it. I mean, if you look at every drug interaction pair, I'm sure we're less than 0.1% of potential interactions being reported. Actually, I'm sure it's much less than that. So what can we do? Well, first of all, we can do what we usually do, create a standardized framework for this incidence evidence generation and dissemination. And we can fill in the gaps, although I would argue that it's mostly gaps. I mean, we're not filling in gaps. We're kind of blanketing the literature for things because it's mostly gaps. And we can do it in a systematic way that's very different from what it is now, where it's a sampling of different approaches that were taken that you try to come up with one number. Next slide. So let's take an example. Let's take one that we know that ACE inhibitors cause angioedema, and we want to measure the incidence of angioedema. And if you ask most doctors, they know that angioedema and cough are the two main things for ACE inhibitors that we think of. But if you ask them, they probably don't know the incidence. Next slide. If you look at the product label, I think it was 0.1%, but I'm not seeing it. There we are. Thank you. Angioedema has been reported in patients receiving lisinopril at 0.1%. Presumably, that's a form of incidence proportion. Next slide. Then if we look in the literature, now these are incidence rates, we see rate per 1,000 person years. So let's take 1 year as our standard. So we're going to have a mixture of incidence rates and incidence proportion. Let's just assume the incidence rate per person year matches the proportion. Like, in other words, on average, and this is a long-term drug. It's not an antibiotic they're getting for 5 days, but a drug they're taking chronically. So I'm going to equate the two of a year-long time at risk. And we see numbers for around 2, around 3, and around 4 in the literature. That's per 1,000 person years, or 0.1, 0.2, 0.3, 0.4%. Next slide. What do we see? If you look in UpToDate, if a clinician goes to a knowledge resource, they're going to see that the estimation is between 0.1 and 0.7%, which matches the previous two slides, if I put them in percents. And now this is just plotting what I've just reported on kind of similar axes, ignoring the fact that they're proportions or rates. They're in the same order of magnitude. Next slide. So now if we run our analysis, so here's an example of running on 8 databases. And you see the upwards, downwards, and upwards triangle. That's that lower and upper bound that I mentioned earlier. And you see rates in the 0.1 to, well, 0.8 at the maximum, more likely 0.5% range gotten from our OHDSI network. Next slide. And if we plot that with the other information we have, the product label 0.1, up to date 0.1 to 0.7, the published studies and our rate of 0.1 to 0.8, they're all in agreement. So we seem to corroborate the literature. Now, I've already stated this, but I'll just restate it. If you go look this resource up and you look up a rate in a drug after starting a drug, the rate could be high because it's causal, or it could be an indication for the drug. So let me give you an example. If I take a statin, I'm going to find patients have a high incidence of heart attacks after it. Why? The statin reduces the risk of heart attack, but the reason they're getting the statin in the first place, the indication is that they're already at high risk for heart attack. Even if the statin works, it'll lower their rate, not to the general population, but to something that remains a little high. So again, when the rates are low, we can say maybe it's not something to worry about. When the rates are high, we have to figure it out. So we actually did our proof of concept in 2017. That was our opening screen for HowOften.org. Next slide. By the way, you'll notice on that slide some warnings, caveats, what this does not do, and then an emphasis on what this does not do. So trying to convey, what an incidence and that it's different from a causal relationship. Now, next slide. So our experience, we opened it up to the world. We mentioned that it was running to the emergency department at New York Presbyterian Hospital, Columbia. It was used by some staff, to be perfectly honest, mainly one person who knew what informatics was. And they tended to use it, but they were actually a senior member of the ED, so they would be looking at many cases, not just the people who came into them. And if someone came in with a bizarre symptom, they would ask what their meds were and would occasionally look in this resource and just see, is this something I should worry about after you're taking this drug? Well, we hadn't done a formal evaluation. So that then became the trigger for Anna Ostropolitz as part of her PhD to do the data consult service. So that was Myriad, Stanford, similar service. But the thing that happened at Columbia that brought it about was the data consult service. And the index question for her service before she actually even went live was a patient who I guess had developed angioedema and they happened to be on penicillin. They asked, is this a likely cause of that? Now, it's idiosyncratic, so it could always be a cause of it, but it's something that we should have thought about ahead of time. And we just looked it up on HowOften.org, and penicillin had a very low rate of angioedema. And so we thought it was unlikely or not something they should have thought of ahead of time. Now, we did some studies, and actually, Elise Ruin will be submitting an OHDSI Symposium abstract this year going over some of the rates. What we found in general was that the rates matched the literature pretty well, but there were some exceptions, and sexual dysfunction was vastly underestimated in OHDSI's EHR data versus studies that had been done more carefully. And then we didn't maintain the proof of concept at that point. Basically, we were busy doing legend studies and other work. Next slide. One way to get something done is to have more help, though. And so we think it's an opportune time to do this. We showed the proof of concept, but we know we need the large network and we need help doing this. There are a number of things that we can do in terms of the phenotype, the incidence rate generation, and how we present the results to users. So the database, so the data itself, the phenotype, the incidence rates, and validating them, and then how we communicate that to people. And so that's kind of where we need help in summary. Next slide. Just to say thank you to the team on the original thing. I hope that this team grows enormously afterwards. Next slide. And I think I hand it over to Patrick now.
Yeah, thanks, George. So George is trying to, hopefully has motivated you to understand that we've done a bunch of work over the last several years that has demonstrated that characterization results are useful, reliable, and potentially can be impactful. And given that this is something that we've now done as a proof of concept across a small collection of databases for a small collection of target cohorts and outcome cohorts, I think now seems an opportune time for us to be thinking about what we as a community could do if we all rallied together around this opportunity for large-scale characterization. So this is in part why at the OHDSI Global Symposium in October, we will be spending a full day together as a community working on this problem. And we really hope that all of you will take this opportunity to engage and participate in this process with us, which is why we've actually dedicated the time at the symposium, that there's no other competing activities going on. This is something that we think everybody can and hopefully should join us in this journey of trying to drive this forward. We're also particularly, George described the local experience at Columbia, but we're particularly motivated by what we observed during the COVID pandemic and the work that the EMA and the FDA required, where everybody seemed to be chasing a background rate. And it kind of, from my personal perspective, it seemed kind of unacceptable that we didn't know what the background rate of myocardial infarction was, that we didn't know what the background rate of deep vein thrombosis was, that we didn't have this information just like readily at our readily at our fingertips to be able to use when critical decisions needed to be made. So I think it's a really good opportunity for us to showcase what we have as a data network, as an analytic community, and to generate reliable information that's that can have a real impact in public health. So what I want to do is to tee up getting you all to think about how you can contribute. I'm just going to just summarize what we are currently thinking about, and I sincerely welcome input and feedback, and we're going to open it up to get your thoughts about this as well. But how George and I have been brainstorming it on a whiteboard about 10 feet from his office right now, scribbled all around, is that, let me back up a half step. George mentioned that when we talk about large scale, usually in OHDSI style, we're not thinking about running one hypothesis or 10 hypotheses, but we're talking about a ridiculous number of hypotheses. As an example of the space that we could consider, there are like 2,000 active ingredients that are, approved by the FDA and in use in a US data set. There's about 5,000 SNOMED terms that have active data in any given database in any given year. So if you were to just say every exposure by every outcome, well, that's 10 million hypotheses, 10 million target outcome pairs for which, in general, we don't have evidence. Now, at some juncture, hopefully in the near future, we're going to be able to develop the analytic capability for us to generate information for all of that. However, what we observed during the COVID pandemic was that there's also a strong pull not to necessarily just do code-based phenotypes, but to instead think about what are the target populations of interest, what are the outcomes of interest, and to think about large scale being a universe of defined phenotypes of targets and outcomes that we may be interested in. So as George and I have been thinking through the process, we've been thinking, wouldn't it be nice if the community's efforts towards developing phenotypes could mature even further beyond, what Aza and Gautam led us on in Phenotype February earlier this year. We have a phenotype library that has several phenotypes populated, but maybe using HowOften as a motivation could encourage many more in our community to contribute phenotypes to this process. And if our phenotype library contained all the phenotypes that we would want to represent both the target cohorts of interest and the outcome cohorts of interest, then we could design an analysis procedure that would just take out of our phenotype library all the phenotypes that we have done and be able to compute the incidence rate for every target times every outcome. Now, when I describe target cohorts, George just featured that initially we've been thinking about drug cohorts, a given population exposed to a but the work that we've done up to this point, we've thought about the general population, if we need a background rate within a given defined population. Within our Odyssey community, we have work groups that are specialized in specific clinical subspecialties. We've done work in diabetes and hypertension. We've got our oncology work group and our psychiatric work group and our vision care work group. And each of them have specific indications or diseased populations that may be worthwhile to explore on their own. We can also think about drug classes. Each of these represent target populations that we can think of as the denominator that would represent any of these incidence rates that we could calculate at scale. We also have a need and desire to think about what are our outcome cohorts that we may be interested in. And as a community, we've made good progress on creating phenotypes for the adverse events of special interest, for the EMA's designated medical events. We've also seen progress in the community creating indication outcomes, as well as side effects of drugs that we may pay attention to. And while we've made progress on phenotyping examples of these target cohorts, outcome cohorts, if you were to look today in our phenotype library, you'd still see lots of gaps. And so we see one big opportunity as a community is for us to use HowOften as the motivation to get each of our work groups who are interested in contributing to contribute phenotypes into the phenotype library as an input to this overall process. Assuming that we do have all those phenotypes done, we can go on to design a protocol and an analysis plan that's going to execute the analysis that George highlighted. And all of those open questions that George presented, those represent things that we need to really be thinking carefully about and making some decisions about as part of this analysis. Now, if we generate evidence across the Odyssey network, and we are talking about millions of hypotheses across dozens of databases with all of these analysis configurations, it means our result set is going to be extremely large. And that means that there's a real opportunity for us to think about how we develop user interface to explore those results. Because it's not just a matter of here's a file with some incidence rates. We actually need to think critically about how we're going to disseminate that evidence in a useful and meaningful way. Now, let's imagine that we had our phenotypes, we had a protocol, we had an R package out there, we even had a draft interface. Ultimately, this project's going to succeed or fail based on having data partners who have access to data in the OMOP Common Data Model to be able to run this package, share results, and that collaboratively we can interpret those results, which I think is going to have three direct implications. One, from a technical perspective and a development perspective, if as we start interpreting results, we may realize that there's more development to be done on that interface. We may be able to quickly iterate on developing more effective tools for exploring this ridiculously massive result set. I think we're also going to find methodologic insights about how to generate reliable evidence and what does it mean to look at incidents across a data network that we certainly think is going to be scholarship that warrants publication. But also, if we are able to produce background incidence rates in specific disease populations with specific outcomes of interest to particular populations, then all of those clinical findings is explicitly filling the evidence gap that George highlighted that we still see is very much present. And so some of this work we believe can be happening between now and the symposium, and some of this work is what we envision doing together as a community at the symposium. But for all of you on the call, I see 130 of you on now, we need your help. This isn't something that the proof of concept was something that a relatively small group participated in, but if we're going to really make this be a serious contribution by our community, we need phenotypers who are going to come together to develop phenotypes for the target cohorts and the outcome cohorts and enter those phenotypes into our Odyssey phenotype library. We need R package developers, those that actually want to be able to take the ideas that we are codifying and contribute to creating an R package that will be able to run across our entire community. We need developers for this user interface. If we're developing an R Shiny app, we need folks who can think about that UX design and implementation. We need data partners who are going to be willing to execute this network study, generate results, and share them. And we need methods researchers who are going to think about this evidence and how it could be reliable and how we can conduct a methodologic experiment to prove that the evidence we're generating should be used meaningfully and appropriately. And we need all of you who are clinical researchers to contribute to interpreting the results and sharing them in the context of what currently is known in the field out there at large. So our next steps, as we are thinking about it, and we welcome input, is before the global symposium, we are going to start the process of drafting a protocol that'll describe what we're doing, and hopefully this will allow for data partners to get approval to participate in this large-scale effort. We need to develop and evaluate phenotypes for targets and outcomes. And I really hope that particularly all of our work groups who have a particular interest in a particular disease subspecialty or a specific population of interest will consider using your work group time as an opportunity to create those phenotypes and contribute them to our phenotype library so we can pull them into the analysis. And we're going to be working towards releasing an analysis package that uses that phenotype library as an input so that we can instantiate the cohorts and run a large-scale incidence package. And that's going to involve development because to date, we have not used any of the incidence packages that exist in our community, whether it be the cohort incidence package that Chris Knoll led the development of, the incidence prevalence package that's been developed by the Darwin EU team. None of these have been implemented at scale. So we don't really know what's going to happen when we run an analysis that maybe contains millions of hypotheses. But this is something we can work on between now and the symposium to figure out. At the symposium, what we're going to hope to happen is that we will be able to continue the conversation about what HowOften is going to contribute to our field, to execute the analysis package with all of our data partners who are there, to deploy an interface that will allow us to explore the results, and for us to be able to collaborate on these methodologic development and clinical questions so that what we've learned from the data can be something that as a community we can push out there. So with that,
Patrick,
I want to...
Patrick?
Yes.
Just a quick comment. You know, if we produce all drugs, all side effects, let people know it's there, if it's reliable, if it's something that people can cite. I mean, our experience in the last 5 years is in working with other groups like the EMA and FDA. I mean, these are rates that they use, that they need, and researchers. How we can get it used in practice at the point of care is also a good question. But I think this is a resource that OHDSI can produce and put its name on that would increase the visibility of OHDSI and actually be useful for people. And when you do a study that does incidence, it should be that, well, I'm going to go and check, you know, how I compare to the OHDSI resource. Like that should just be the standard thing everybody thinks of going forward. So I think there's something, I think we can benefit healthcare by doing this, and I think this can benefit OHDSI. The first version, the phenotypes were just SNOMED codes, you know, and their descendants. And now we're looking to do stronger phenotypes. Where do we end up? I don't know. It could be that these phenotypes are what we use to evaluate our phenotypes using SNOMED codes. It could be we do the thousands of phenotypes we need manually and then end up using them to replace the SNOMED codes. Or that we have an effective way of showing the user, look, this is a validated phenotype versus just a SNOMED code, and we can convey to them the difference between the two. So if it's something no one's ever cared about before, yeah, you're stuck with the SNOMED codes. But if it's something like myocardial infarction or kidney failure or something, that's going to be a stronger phenotype. So that's all part of, you know, this design process to figure out how we should do this. Thank you, Patrick.
Yeah, no, that's great, George. Thanks. So we've got 10 minutes and we purposely want to collect some input from all of you. So in addition to using the chat, I've also opened up pollev.com/Patrick Ryan 800. And we're going to have 2 questions and we really sincerely want to hear from you. So now what you've heard is we have a pretty audacious goal here, which is figure out a way to define the universe of all target cohorts and all outcome cohorts and produce incidence across our entire network and make that evidence available. That's going to start with trying to define what are those target and outcome cohorts. So we'd like to take a few minutes, if you can go to pollev.com/Patrick Ryan 800 and enter in what phenotypes do you think should be included in this set. There's diseases that you're interested in, exposures, or other populations. We'd like to hear that. And hopefully, as you're entering those in with your ideas, these can become targets that our community can go after as phenotyping opportunities. And in particular, I want to encourage all of our work groups to be thinking about if you have some selfish interest. I'm thinking about Sally and Cindy in the vision care group. Like if you have a selfish interest in, I just want to study blinding diseases. Well, this is a great opportunity for that work group to create a phenotype for macular edema and age-related macular degeneration, and create those phenotypes, and those can become populations. Or Andrew and Deem in the psychiatry work group. This is the chance to create a phenotype for depression and bipolar disease, bipolar disorder and schizophrenia. Or Asya and the entire team in the oncology group. This is the chance to create a phenotype for breast cancer and lung cancer and prostate cancer. But those populations can both be used as target cohorts, representing populations that are the denominator for which we want to look at the background rate within that population. And they could also be outcomes. So we can think about within a population, how often do you actually observe breast cancer, lung cancer, prostate cancer? And these are very fundamental pieces of information that if only we had a international global resource across all of these questions that we could make accessible. While you guys are entering in, I see that Scott posted a comment in the chat. So since there will be ongoing phenotyping, is there a thought that there will run additional runs periodically? If we are working towards a phenotype library, what happens if there are several valid phenotypes, each with a different purpose? Is there some thought about characterizing? Oh, George just responded, but I'm going to just finish this up. Is there some thought to characterize these and be able to explain the difference, the quality, etc.? So I think, Scott, you're spot on. What I'm kind of hoping for personally is that we're using HowOften as the kick in the butt to get everybody to contribute phenotypes. We've now talked about phenotypes now for a couple of years, but phenotyping isn't the end game. And getting a phenotype into the phenotype library isn't the end game. It's the use of the phenotypes to generate reliable evidence that's important. And so very much we're using this as an excuse to kick something off that we hope will become a sustainable process. Imagine we can create a computable process that just takes in phenotypes and computes these things, then that could become a living resource, a process that we run over and over again, and we can continue to expand our phenotypes, continue to expand the horizons of what we're computing. But we want to use this as a big effort to try it once to see how far we can get if we all collaborate together. Right. I'm seeing lots of entries here for, we've got some COVID-related things, we've got cancers, chemotherapy toxicities. Lots of people excited about cancer, rheumatic diseases, HIV, glaucoma, opportunistic infections, death, diabetes, fatigue syndrome. Looks like our oncology work groups got their work cut out for them because they've got a lot of interest in creating lots of oncology phenotypes. It'd be wonderful to see those entered into our phenotype library. We've got depression, autoimmune diseases, Down syndrome, type 1 diabetes. So there's a wide array of erectile dysfunction, myocardial infarction. So there's a wide array of phenotype targets. If you look right now in the OHDSI phenotype library and you don't see the disease that you're interested in, and you're thinking, geez, I sure hope that that phenotype will make its way into howoften.org, well, this is your chance. Grab that phenotype, follow the processes that Aza and Gautam have outlined, join the phenotype work group, and share the progress that you're making. We've defined over and over again how to create a phenotype. You write a clinical description, create some concept sets, create a cohort definition, evaluate it using OHDSI standard tools like cohort diagnostics and p-evaluator. That's something that our community can be collaborating on together, and you could start now. There's no excuse or there's no reason to be waiting, particularly now knowing that if you do that work, that can feed nicely into this larger effort that George is trying to spearhead here. All right, see lots of, I'll let you, you're still entering things in, so which is good. We got one more question, so I am going to flip over to it to switch gears. So George and I are very cognizant of the fact that we think this would be a good resource, and appreciated that Thamir entered in the chat there that it also would be a big need to know these rates. But we'd really like for you also to be thinking about critically why you think HowOften might be useful. And so we want to hear from you. What are the novel ways that you think HowOften might be used by stakeholders? So we've seen, for example, we've seen regulators use background rates computed from the OHDSI community to contextualize spontaneous adverse event reports. We've seen examples, as George highlighted at Columbia, where understanding that an event rate is low is sufficient to determine whether or not that outcome should be considered in the benefit risk calculus of a particular product. But we also, as George and I have been brainstorming this for quite some time, we also kind of feel like we don't know all the potential use cases of this resource. That if the world had a resource that had the incidence rate of every target cohort by every outcome cohort stratified by age and sex and index year, we think that there's a lot of utility to that, but we don't even know all the possible uses of that. And so we would love to have the collective minds in this community thinking about if this resource or when this resource is created and made available, what could we do with it? And so would welcome and would love to hear from you about the different use cases that you all anticipate could take advantage of this resource. So we're seeing inspiring future causal questions. And exactly as George highlighted, this is not a causal inference, but it might help you understand which question you might want to go after to determine what that attributable risk is. Check it before running a study. So, can it be used as a resource just for feasibility assessment? Important for clinicians just to understand the therapy that they're giving to their patients. As Thamir highlighted, it's oftentimes the case that we don't even know how often side effects happen. So you just give somebody a product label with a rattled on list of 50 adverse events with no context for whether they're going to happen a lot or a little. And providing that information could be really helpful. Check to see how well our phenotypes compare to other phenotypes or other health systems. So indeed, a big part of this is going to be trying to contextualize measurement error, and this could be a mechanism to help us understand that. Validation and input to digital quality measure development. So certainly this could be a good starting point for understanding that. So I want to encourage, as you're all thinking about this and entering your thoughts here, I would really welcome and encourage all of you to think about participating in this effort. And George and I are really trying to start a big boulder rolling here. We think that this is a great idea. We are really excited about driving this in the community, but there's the only way it's going to happen is if everybody jumps on and helps us push this boulder as well. So over the next couple months, as we're getting ready for the symposium, encourage those of you who are interested to start contributing phenotypes to help us with the development of the analysis package. And we'll be posting as we continue to develop the protocol. And for those of you who are coming to the Global Symposium or thinking about coming to the Global Symposium, really hope that you'll join us for the entire day, whether you be a data partner or a clinical researcher or a methodologist, and be eager to join in this effort as we try to advance this idea. I know we're at the top of the hour. George, anything else that you'd like to highlight?
No, just thank you.
Great.
It'll be fun.
Thank you all very much for coming today. Appreciate it. I look forward to seeing you all next week and look forward to collaborating with you all on HowOften moving forward. Thanks, everybody.